Hi there, this is the first part of phylogenetics, and you can find the information in chapter 24 of the Integrated Molecular Evolution, second edition. The definition of phylogenetics is the study of the pattern of evolutionary history, and it comes from the Greek roots phylon and genetikos. Phylogenetic reconstruction is the determination of genealogical history of specific lineages, and these can be individuals, your family members, for instance, species, genes, or whatever you want to use, whatever data you want to use that indicates genealogical history. Cladistics, on the other hand, is the grouping of organisms based on shared characters. Now, while they can be molecular characters, more often they're morphological characters. So this doesn't always indicate genealogical history. So it's mainly a grouping of organisms based on their shared morphological or other characters. Phylogenetics is based on the principle that there are sequential, predictable, and continual mutations that occur in DNA sequences, or RNA or protein sequences as well, some of which are passed on to RNA and or proteins through transcription and translation. The rates of change and mutation, as well as selective pressures, determine the ultimate rates of mutation that can be observed in those DNA, RNA, or protein sequences. Most methods use calculations based on the minimum number of mutations, often called minimum evolution. So, what kind of questions can be answered using phylogenetic methods? One is answering what taxon or gene is this. It can group with other similar taxa and genes. How is this taxon or gene related to other taxa and genes? That is, how far is a human from an E. coli or an elephant or whatever you want? Is the gene a member of a multi-gene family? So these would group together as a multi-gene family. What is the origin of the taxon or the gene? So you can get back to where these originated or diverged from other species. Has the gene been inherited vertically or horizontally? And this can be determined by comparing genes that are inherited vertically, and then look at any that appear to be to give a different pattern indicating horizontal origin of those sequences. What is the origin of the organelle or endosymbiont? And this can often be done. These are pretty interesting, actually. And what is the rate of mutation or evolution in this gene or multi-gene family, or whatever's being measured? Here's the first phylogenetic tree that we know of. It was drawn by Charles Darwin in one of his notebooks in 1837 after his trips or his voyage on the Beagle. And you can see that above this he wrote, actually, I think, and he was indicating that species would diverge into other species along a genealogical line. By 1879, the idea of the genealogy of all these species into one unifying tree was fairly well accepted. So Ernst Haeckel drew this one, and he drew many other trees as well. And you can see it starts with the protozoans, very simple organisms, but still eukaryotes. And then going up into the vertebrates, including the age of fishes, when you have many divergent species of fish. And then the age of reptiles, and that's primarily dinosaurs and things like that, and birds branched off of one of those lines. And then into the age of mammals from the Eocene onward. By the mid-20th century, other trees were being drawn just to distinguish among the various groups of organisms. So in Whitaker's tree in 1969, there was a plant line, there was a fungal line, and there was a animal line based on photosynthesis, absorption, and ingestion of food. Monera down below with the bacteria, then diverging into the protists down below. So a little more sophisticated than the earlier trees, but still not quite what we have at the present. By the 2000s, then, we have a tree that looks like this, and this one is sort of bacterial-centric. So the bacterial groups are accentuated in this one, going from the firmicutes, Chlamydia, actinobacteria, and proteobacteria, etc. And then over into the archaea here, and then the eukaryotes. So in this case, it's a circular tree. This is so that you can get more species names on these particular trees. And then with the advent of more DNA sequencing, here's a subsequent tree where now the excavata, alveolata, chromista, rhizaria have been split out from some of the other groups. The fungi are clearly now up closer to animals in the what's called the Opisthokonta, and then amoebozoa coming off of that, and later plants. And then this one's a slightly more recent tree, especially concentrating on this group, the excavata. Members of this one group are photosynthetic, including euglena, whereas the sister group to that includes trypanosomes, which now aren't photosynthetic. And then Hacrobians have been split off. The chromista have really been redefined as Stramenophiles. Here's the alveolata and rhizaria. And other than that, the other groups are fairly similar. Plants, amoebae, and Opisthokontas, which includes animals and fungi. As I said, phylogenetics can be used to look at the relationship between some unknowns and some knowns. What taxon is this and how it relates to others. So these are some isolates, the ones that say GI. Those are glacial isolates that we isolated from various ice cores around the polar regions. And then they've been put into a sequence alignment and then compared phylogenetically with other species that are from polar regions as well. So in this case, everything in black font, those are the glacial isolates, things that were grown in culture, fungi that were grown in culture, and we wanted to compare that with fungi from known sources, the red from soil, the green from deeper soil, the blue are from aquatic and marine sources. And the light blue, there's a few light blue, are true psychrophiles. The others are mainly psychrotolerant. That is, they can grow in cold temperatures, but can grow in slightly warmer temperatures as well. So the glacial isolates, in most cases, grouped with organisms from either cold water or cold soils from Antarctica or the Arctic. And it solidified our conclusions that these isolates that we had grown from ice cores from Antarctica were authentic cultures from that ice. Here is another example of how phylogenetics can be used. I discussed this phylogram earlier in this semester, looking at the evolution of the alpha globin genes and the beta globin gene family, as well as myoglobin. And so you can see in the evolution from invertebrates to fish, which occurred over 500 million years ago, there was a divergence between the alpha type and the beta type, and you can actually still see that in fish genomes when you sequence these genes. Then around 200 million years ago, there was the evolution of mammals for the first time, and so there was a split between versions of the alpha globins and the beta globins. And finally, about 100 million years ago, there was a further divergence between placental mammals and marsupial mammals. which cause divergence of more duplication and divergence of more alpha and beta globin genes due to the need to transfer the oxygen from the embryo to the fetus and to the adult, essentially. And so you can actually trace those changes in those genes with those evolutionary developments of the various types of vertebrate organisms in this case. You can also trace primate evolution, including the evolution of humans down here. And so you can see going from the lorises to the lemurs to the tarsiers, and then eventually to apes and monkeys down at the bottom, including humans. You can trace those lineages using phylogenetic analyses, and you can compare that to a tree based on fossil records on the left of the various organisms. First the Australopithecus, and then the Homo species going through Homo erectus, Homo neanderthalensis, heidelbergensis, and eventually Homo sapiens at the top. So frequently the molecular data used for the phylogenetic analyses are compared with fossil records, morphological records, things like that, to confirm the separations of the different organisms, the relationships between the different organisms, and the diversity of those groups. This tree was determined back a few decades now based on human mitochondrial DNA. So it's essentially tracing what was called at that time the genetic Eve. And the deepest branches from the ancestor there, which occurred around 200,000 years ago, are from Western Africans and also Eastern Africans. So pretty much Central Africa, spreading out from there. And then other groups going into different areas of Africa, primarily Africa, a few Asian lineages in there also, very ancient Asian lineages. And as you continue on around the circle, it's mainly from Africans, and finally more Asians, a few Europeans, a few more Europeans, and then back up to Asian and New Guineans. And so this sort of not only indicated that the original ancestors of humans were from Central Africa, but also traced the spread of that mitochondrial DNA throughout the world. This was from another study looking at not only the origins of humans, but also trying to identify where some of these migrations occurred. So you can see things began in Central Africa, and there are various mitochondrial lineages indicating diversification of the African population. And then moving over into Asia and all the way through the middle of Asia and further on. And then there was a lineage that went down into Australia, and then a later lineage up into Europe, and then a very late lineage that went down through North America and eventually South America. In that case, losing diversity as it moved into South America. So with this and some other data, maps were drawn looking at the mitochondrial haplotypes, that is the mitochondrial DNA coming from the females, and how it moved through the world, human migration. So 200,000 years ago, the origin in Central Africa. By 170 to 130,000 years ago, spreading through different areas of Africa, and then moving up and out through India and Australia, by about 70 to 60,000 years ago. And then one migration into Europe about 40 to 50,000 years ago. Migrations further into Eastern Asia 35 to 25,000 years ago, and then migrations into the Americas from about 7 to 15,000 years ago. The dotted line indicates that there probably was another migration from Europe into North America by a different route, not the land route, but by a sea route. After tracing the routes of migration throughout Africa and the rest of the world using mitochondrial DNA, other studies were done tracing the Y chromosome sequences throughout the world. And what was found was, starting in Africa, there's pretty much movement of the males and the female markers, the mitochondrial and Y chromosome together, and even up over into Asia and Australia and into Europe. There's pretty much concordance between the mitochondrial and Y chromosome migration routes. Some of the other routes, however, seem a little bit varied as far as where the male markers moved and where the female markers moved, and this might indicate some of the times when there were warring tribes and there were mass migrations and exploration events that mainly the men were going out exploring and the women were staying put. So then there were some differences between the movements and migrations of the mitochondrial and the Y chromosome. The hybridization events could also affect this because some of those markers are neither on the mitochondrial nor the Y chromosome, so that would confuse things a bit as well. When trees are presented, they're usually presented with straight lines joining the different taxa together and then branching in different patterns. And that the lines are pretty linear and just two-dimensional. But if you look at an actual tree and try to identify all the organisms and all individuals within those various lines, they have another dimension, and that is that there's a number of organisms in there that are participating in reproduction and eventual evolution along that line. So in each case, you can have things like a genetic bottleneck where the number of individuals reduces, and this appears to have happened in the human population about 200,000 years ago, that we almost went extinct. You can also have branches where the individuals separate for a while, come back together, and then you get these individuals that have diverged somewhat coming back into meet the other population that has also diverged a bit. And this might happen in those times that where there's migrations of several different groups. They diverge and they migrate in different areas and diverge a bit, and then come back together at a later time when those populations grow together. You can also have these hybridization events, which have occurred in humans, where you have two species that have diverged sometime in the past, and then they can still mate to form hybrids, bringing those sets of genes together into essentially a new organism or a new species. So these branches really represent many different individuals diverging, coming together, and especially in sexual organisms, moving around alleles and genes from one part of the branch to the other. So they're not really solid lines at all, but we represent those as solid lines. And when representing a phylogenetic tree, it's really an inferred phylogenetic tree. So this is a tree of life with bacteria, eukaryotes, and archaea here. But the data that are used to make this tree look like this. So they're out on the tips of those branches. So we take extant sequences from extant organisms extract the DNA, line those up in an alignment to get the genes lined up, and then join those together into an inferred phylogenetic tree. So now we have the branches joined together according to how similar the sequences are, joining them together back into the past. And even then, when you look at each one of these branches, you find that there's a netted pattern to the paths in evolution, so that genes or whole genomes in some cases are being exchanged among individuals and different lines along those trees. And not only that, but in many cases, entire organisms have been joined in those symbiotic relationships with other organisms to create things like mitochondria, chloroplasts, and other organelles. So before we go into the methods, I need to talk a bit about tree terminology. So here's just a basic phylogenetic tree. And the names that are used on these trees are similar to a physical tree. So there's a root, and then there are nodes where branches separate, and there are the branches. The internal branches are known as internodes, and then there are terminal branches on the end. And on each of the ends, then you have the operational taxonomic units called OTUs. So there's operational taxonomic unit A, B, C, D, and E. And if you happen to have the genus and species name or other names for the genes, etc., those become those names at the ends. Trees can come in a couple different styles or forms. First are rectangular cladograms and then rectangular phylograms. Note that the branches are of different lengths. So in a rectangular cladogram, usually everything is lined up along the right-hand side so that all the taxa and taxa names are out to the right side. And the branch lengths aren't accurate. They aren't accurate representations of how distant those taxa are. In a rectangular phylogram, however, the lengths of the branches are meaningful from side to side. And so the lengths from the node to A or the node to B are real distances from those, so that the distance from A to B is a real distance. Also, for rectangular cladograms and phylograms, the vertical lines are simply there to join things together, and they don't represent any sort of distance. Only the horizontal lines represent the distance, and especially in rectangular phylograms, the horizontal lines represent the true distance in nucleotide differences or amino acid differences, etc. On the bottom, then, there are slanted cladograms and slanted phylograms, and in this case, again, for the cladogram, the distances are not, the line lengths are not representative of the true distances between those taxa. However, on a slanted phylogram, those lines do represent the true distance between the taxa, the nodes, etc., based on the nucleotide differences or the amino acid differences between all of them. So for these slanted phylograms, all of the lines, the lengths of the lines are meaningful as far as distances between the taxa and the nodes. Trees can be unrooted if you don't know where the root is for a particular tree, or they can be rooted, but if you do root them, then you should know exactly where that root should be. Otherwise, the relationships can be in error. So in the first case, if you take that unrooted tree at the top and pick it up, just pinch it and pick it up on that branch between the node and B, then the rooted tree would look like this on the left. However, if you pick it up on that middle branch, then the tree would look like this. Now, while the distances between each one of the taxa is still the same. The relationship to the root, and therefore the evolutionary direction, can change significantly and can be either correct or very incorrect. For any particular tree, there is free rotation around each of the branches. So in the top case, you can rotate A to B to B, B to A. You can rotate that DC branch, you can rotate the DCE branch, and you can rotate the whole tree. So as long as the distances have not changed, the tree is really the same, but you've rotated some of them in the tree, and it's really the same tree. When choosing sequences to analyze, when you're looking for taxonomic distinction, when you're looking for gene distinctions, then you have to choose regions to the appropriate level. So as far as conserved coding regions, they change at a substitution rate of about 10 to the minus 6. That means one nucleotide out of a million changes over a certain amount of time, and in this case, per year. As you go to moderately conserved coding regions, the variation is from, or the substitution rate is from about 10 to the minus 5th to 10 to the minus 6th per locus per year. So that's about one change in 100,000 to one change in a million. Introns change a little faster, between about 3 times 10 to the 4th to about 2 times 10 to the 5th, somewhere in that range. Flanking regions and pseudogenes change at about the same rate. But microsatellites and mini-satellites change much, much faster. In fact, you can record differences in your own body, in your cells, that have changed during your lifetime. So they're changing much, much more rapidly. So if you want to distinguish between an E. coli and a human, you'd use conserved genes. If you want to determine differences between you and me, then you would use microsatellite or mini-satellite sequences. And you can make really fine distinctions between individuals that way. Here's one example of how to select certain regions for various types of studies, levels of taxonomic study. So if you look at just the ribosomal RNA gene locus, it starts with an intergenic spacer, which is highly variable, and then a promoter region, which is less variable, and then the genes, the small subunit, 5.8S, and large subunit, and then another IGS after that. If you look relative to all organisms, then the small subunit and the large subunit, especially some parts, are highly conserved, whereas other regions in black are quite variable and can't be used at broad taxonomic ranges. If you look at that closer and look at just the ITS region, then you have regions that vary little at the species level, interspecific variation, and you can use those to determine species. And then if you go out to the generic or genus level, you can see there's more variability. So some regions would be useful at the genus level, and other regions would vary too rapidly to use at the genus level. So I made this chart just as a general guideline to determine, for a specific locus, what taxonomic level can that be used at. So for microsatellites and mini-satellites, they're highly variable. They represent recent changes, and they can be used to distinguish from one tissue to the next, or one individual to the next. At the intermediate level, then, internal spacers, introns, globins can be used at the species to family levels. And then as you go to more conserved regions, those represent ancient changes usually. You can work at the phylum or kingdom level with those regions. Another consideration in phylogenetics is that you're relying on present data, as I mentioned earlier, and then you're inferring back into the past. So you're joining those lines, but really you're basing it on data that you have collected recently from extant individuals. So while the branches and the branching pattern is fairly solidified close to the top of this phylogram, as you work your way further and further into the past, then those connections become more and more tenuous. That is, they become a little less certain as to where the branch and the nodes should be. However, in my lab and other labs around the world, we've been able to isolate DNA from ancient materials, sometimes millions of years old. And so this allows you to actually go back and confirm some of these branches in the past, although it's not simple to do, and often in ancient samples they've deteriorated to some extent, so the sequences aren't as reliable as with fresh material. Another thing to keep in mind when you're dealing with evolution and building phylogenetic trees is that evolution isn't constant. It isn't a smooth line that it's made out to be sometimes. So that instead of following a straight line like the dashed line in A, there's what's called punctuated evolution. So there's periods of rapid evolution followed by stasis, followed by moderate evolution, followed by increase in evolutionary rate, and stasis, and so it goes up in a jagged line. So when we're drawing phylogenetic trees, we're assuming that it's going as a straight line, whereas in reality it might be quite different than that. Another thing to keep in mind is that not all mutations can be observed. So if you start with this ancestral sequence and then have these changes occurring in these other two modern sequences, single substitutions can be measured, right? So you can see this one, a C to an A change. If that's all that happened, that can certainly be measured. However, if there are multiple substitutions, such as a G to C to T change, then two of those changes are missed. So the G to a C and then a C to a T change, those are missed, and it's assumed that there's just been a G to a T change. You can also have coincidental substitutions, a T to a G and a T to an A over here, and the T is completely missed. It looks like instead a G to an A change. Also, there can be parallel substitutions, and in that case it looks like nothing has changed, when in reality both sequences have changed from a C to an A. And then there are convergent substitutions, where in one case there's been an A to C to T change. In another case there's an A to T change, and all that's noted is an A to T change in both, and those have been missed. And finally, in the case of a back substitution, that is a C to a T to a C change, often those are completely missed because it seems like the C is there, and in the other one the C is there and no changes have occurred. So when analyzing sequences, realize, and this is inferred in minimum evolution too, that many of the changes cannot be observed, or for one reason or another. And so you're dealing with the minimum number of changes that lead to that branch or that branching pattern. Okay, that's the end of this presentation. Next time will be part 2 of phylogenetics, where I will talk about specific methods used in phylogenetic analyses. So I'll see you next time.